research colleagues all the best. Go ahead and have a great day. Thank you.
Thank you, Dr. Sukant. We now begin the theme 3, Foundations of Neuroscience, Neural Systems, Disorders, and Emerging Insights, where today's lectures will explore the fundamentals of brain function, neural systems, and their relevance to neurological disorders. Our first lecture will be delivered by Kompala Jhansi, currently pursuing a Bachelor of Engineering in Bioinformatics. Her lecture is titled Understanding the Brain is Understanding Ourselves. Welcome, Jhansi.
Good evening, everyone. I'm Jansi, and today I want to take you on a journey into something that is not just a part of your body, but the very thing that defines who you are, the human brain. Before we begin, I want you to pause for a moment and think about something simple but interesting. Why is that you can remember a random moment from your childhood that can be a smell, a place, or a person so clearly, but at the same time, you forget what you have just studied yesterday? Or why does a particular song suddenly make you feel very emotional even if you don't consciously try to feel it that way? These are not just random experiences, these are carefully processed outcomes created by your brain. The brain is not like any other organ. Your heart pumps blood, your lungs help you breathe, but your brain does something much deeper. It creates your identity. It decides how you think, how you react, how you interpret the world around you, and even how you perceive the reality itself. In a way, every experience you have is filtered through your brain, meaning each one of us is living in a slightly different versions of reality. Today, we are going to explore three powerful ideas. First, how the brain is built, its structure and components. Second, how it works, how signals move and systems interact. And third, what happens when things go wrong and how the modern science is trying to repair and even enhance the brain function. As we go through this lecture, I want you to keep one question in your mind, and I really want you to think about it deeply. If we fully understand the brain one day, does that mean we truly understand ourselves? Or is there still something beyond biology that defines us? Let's now try to understand the scale of what we are dealing with. Because the brain is almost impossible to imagine in terms of complexity. The human brain contains approximately 86 billion neurons. Each of these neurons is capable of forming thousands of connections with other neurons, resulting in nearly 100 trillion synapses. Now, let me give you a simple way to visualize this. Imagine each neuron as a person. Now, imagine each connection as a conversation between two people. The number of such conversations happening inside your brain at any moment is far greater than all the conversations happening on the entire internet combined. That's how dense and interconnected the system is. But here is something even more fascinating. Your brain processes about 11 million bytes of information every second. However, your conscious mind, that what you are actually aware of, it can handle only about 50 bytes per second. That means more than 99.99% of brain activity is happening without your awareness. For example, right now, without you thinking about this, your brain is controlling your breathing, maintaining your posture, adjusting your body temperature, filtering the background noises, and helping you to focus on my
मी पण जातो.
voice. You're not consciously telling your body to do any of this. It just happens. And despite all our scientific progress, we have only mapped about 1% of the brain in detail. That means most of the brain remains unexplored, like a universe. We are just beginning to understand this. So, let me ask you, do you think it is even possible for humans to fully understand something as complex as the brain? Or will there always be some mystery left behind? Before we go deeper, let me guide you through what we are going to cover today so you can follow the flow easily. Think of this lecture as a journey. We will start at the smallest level, the building blocks of the brain, and then slowly move to larger systems, then to disorders, and finally to the future of neuroscience. It's similar to understanding a city. First, you look at the individual buildings, then the roads connecting them, and then what happens when something breaks, and finally, how the technology can improve the system. And here, first, we are going to look at neural architecture, which is how the brain is built with neurons, synapses, glia, and myelin sheath. Secondly, the brain function, how the brain functions, how the brain systems like neural system, regions, and the neurotransmitters function. Third, neurological disorders. When something breaks, what it leads to, the neurodegeneration, epilepsy, and psychiatric conditions. Then, the diagnostic tools. How can we look inside the neurological disorders? We can have these diagnostic tools like MRI, fMRI, EEG, PET, and emerging biomarkers. Next, emerging research. Where it's headed, like AI, gene therapy, BCIs, and psychedelic medicine. At last, the future scope. Neuroscience and its future across the medicine, technology, and research. By the end of this session, you will not just know the facts, you will understand how the brain functions as an integrated system. And I want you to involve you here. Which part excites you more? Understanding diseases like Alzheimer's and Parkinson's, or exploring the future technologies like brain computer interfaces? Now let's begin with the foundation, the building blocks of the brain. The most important cell is neuron. Neurons are highly specialized cells designed to transmit the information. Every thought, every movement, every memory, and every emotion begins with the activity of neurons. What makes neurons remarkable is not just their number, but their connectivity. Each neuron can connect to thousands of other neurons, forming a massive communication network. A neuron typically has three main parts: dendrites, which receive the incoming signals, the cell body, which processes the information, and the axon, which sends signals forward to other neurons. Now, communication between neurons happens at structures called synapses. At these junctions, signals are passed either through chemical messengers like neurotransmitters or through direct electrical connections. This constant communication is what allows your brain to process the information rapidly. Alongside neurons, we have glial cells. For a long time, scientists underestimated their importance, thinking they were just support cells. But now we know they are essential for maintaining the environment of the brain, like protecting neurons and even influencing communication. Another critical component is the myelin sheath. This acts like insulation around electrical wires. It allows the signals to travel much faster and more efficient. Without myelin sheath, communication becomes slow and unreliable. And this is also a fatty insulating layer around the axons that accelerates the signal conduction up to 100x. For example, in a disease like multiple sclerosis, the myelin sheath is damaged. As a result, signals cannot travel properly, leading to the symptoms like muscle weakness, coordination problems, and vision issues sometimes. So, if you think about it carefully, your brain is like an advanced electrical network. But unlike machines, it is constantly adapting, learning, and reorganizing itself. Now, think about this. What would happen if your brain suddenly slows down drastically? Even basic actions like speaking, walking, or reacting would become extremely difficult. Now, let's talk about how neurons communicate. They usually use chemicals called neurotransmitters, which are also known as the brain's messengers. These chemicals influence your mood, behavior, and your thoughts. Neurotransmitters are the chemical language of the brain. Imbalances in these molecules underlie many of the most neurological and psychiatric disorders. And the more neurological drugs work by targeting these pathways. For example, dopamine is linked to reward and motivation. When you achieve something, the dopamine is released and it instantly makes you feel good. For example, when we are publishing a paper and we are receiving a positive feedback in the first review itself, and then the dopamine is released and it actually makes you feel good. Second, serotonin, which regulates the mood and sleep. Low levels are associated with depression. The low levels in serotonin instantly leads to depression. Third, GABA, which primary role is inhibition and calming effect. GABA has a calming effect, while glutamate is involved in learning and memory. Acetylcholine plays a role in muscle development and memory. So, your emotions and behaviors are not just abstract experiences, they are linked to chemical changes in your brain. So, that's why meditations for mental health conditions often target these neurotransmitters. Now, think about this. Why do you feel better after an exercise or listening to music or going for a morning walk? Because these activities influence your brain chemistry and they regulate the neurotransmitters to function their way. That is why you feel better after doing an exercise or listening to music. Now, let's talk about what happens when the brain doesn't function properly. What if the brain breaks down? Neurological disorders affect millions of people worldwide, and neurological disorders represent one of the greatest global health challenges. According to World Health Organization 2023, 1 in 6 people worldwide is affected by a neurological condition, making awareness and early intervention critical. And these are of three types. First, neurodegenerative diseases. For example, Alzheimer's disease, Parkinson's disease, ALS, and Huntington's disease. Secondly, electrical and structural, which includes epilepsy, brain stroke, traumatic brain injury, and brain tumors. Third, developmental and psychiatric disorders. For example, autism spectrum disorder, schizophrenia, ADHD, depression, and anxiety. There are two giants of neurodegeneration. First is Alzheimer's disease, second is Parkinson's. Now let's look into Alzheimer's disease, which is caused by amyloid plaques and tau angles, and it is also caused by the extreme accumulation of protein in the brain, which includes the progressive loss of memory and cognition. Till now, 55 million people are affected globally to Alzheimer's disease. There is no cure to Alzheimer's, there's only symptom management. That is why early detection is critical. Here, early detection via PET scans and CSF biomarkers is transforming the outcomes. Next, Parkinson's disease, which is caused by the loss of dopamine in the substantia nigra. And the symptoms of Parkinson's disease are tremors, rigidity, and bradykinesia. Till now, 10 million people affected globally to Parkinson's, and this can be treated with a therapy and the emerging gene therapies and deep brain stimulations offers a new hope to cure Parkinson's disease. While the both conditions underscore why early detection is everything, the window for intervention is narrow but powerful. Now the question is, how can we see inside the brain? Modern neuroscience relies on powerful toolkit of imaging and diagnostic technologies, each revealing a different dimension of brain structure and function. So the tools include MRI, fMRI, EEG, PET scan, CT scan, lumbar puncture. So firstly, MRI, which is also known as magnetic resonance imaging. And what it reveals the high resolution brain structure, detecting the brain lesions for multiple sclerosis, and it also helps in imaging joint ligament tears like knees or shoulders and identifying the soft tissue tumors. And the key applications of MRI is that the structural diagnosis and tumor mapping. Secondly, fMRI, functional MRI, which measures the blood flow changes to map active brain areas while a person performs task. For example, identifying the language or motor center prior to the brain surgery, or studying the brain activation during cognitive tasks, and analyzing the brain activity changes in neurodegenerative diseases. And its key application is functional mapping and research. Thirdly, EEG, which is a non imaging test that records the electrical activity of the brain. And its key applications are epilepsy and sleep disorders. Next, PET scan, which is also called as positron emission tomography scan, and it detects the metabolic or chemical changes in tissues to find the diseases before structural changes may appear. So it helps in identifying metastatic cancer cells, locating the source of seizures, mapping the amyloid plaques in Alzheimer's disease, and checking heart muscle viability after an heart attack. So its key applications are Alzheimer's, cancer, and epilepsy. Next is CT scan, which is also called as computed tomography scan. It uses X-rays to create cross-sectional images, and it is ideal for fast, detailed bone and tissue imaging. For example, it helps in diagnosing head injuries and diagnosing pneumonia or lung infections, identifying the complex bone fractures, and assessing the abdominal emergencies like appendicitis. So, the CT scan applications are trauma, stroke, and head injuries like hemorrhage. And at last, lumbar puncture, and it reveals the cerebrospinal fluid biomarkers. And its key applications are Alzheimer's, MS, and infections. There is an emerging breakthrough in 2024 to 2025. Blood-based biomarkers for early Alzheimer's detection are transforming the diagnosis by enabling non-invasive, accessible screening years before the symptoms appear. And this is a major discovery in Alzheimer's detection. Let's move on to the future of neuroscience. Where neuroscience is heading in the next decade, especially how technologies, biology, and AI are merging. This consists of 6 parts. First one is neuroimaging AI. Here, the AI and machine learning models are now being trained on brain scans like MRI, fMRI, and CT scans. These neuroimaging AI systems can detect the diseases like Alzheimer's, Parkinson's, and tumors earlier than they help in identifying the subtle patterns that are invisible to radiologists. And they can also improve the diagnosis accuracy. And some reports shows 94% plus accuracy. And the big shift in the neuroimaging AI is that diagnosis is becoming predictive, not just reactive. While the second part, gene therapy, where CRISPR allows the scientists causing the neurological disorders, and the scientists target conditions like Huntington's, ALS, or inherited brain disorders. Gene therapy is potentially a cure, not just a treat. And the future impact of gene therapy is that one time genetic fixes instead of lifelong medication. And the third part, BCIs, which are brain computer interfaces. The companies like Neuralink, and the projects like BrainGate are working on connecting the brain directly to the computers. And this BCIs helps the paralyzed patients move their robotic limbs or type with their thoughts. And sometimes they can also restore vision or speech in some cases. And the long term vision of BCIs is that humans interacting with machines using just their mind. And the fourth part, psychedelic assisted therapy, where the substance like ketamine are being re-evaluated scientifically. And these are used for treatment resistant depression, PTSD, and anxiety. These psychedelic assisted therapies helps reset the brain connectivity and emotional processing. And some have received breakthrough therapy status by regulators. And the key idea of psychedelic assisted therapy is that the mental health treatment is moving beyond the traditional drugs. And the fifth part is brain organoids. These are the tiny lab grown brain like structures made from the stem cells. These brain organoids mimic real brain tissue at a small scale, and these are particularly used for drug testing and disease modeling. And they can also be helpful in studying the disorders like autism, epilepsy, and even neurodegeneration. The advantage of these brain organoids is that ethical alternatives to human or animal testing. These brain organoids are used for ethical alternative to human or animal testing instead of trying it on a mice or any animal. In the sixth part, we can see the connectome mapping. The projects like Human Connectome Project aim to map every neural connection in the brain and understand how the circuits control behavior, memories, and the disease. Think of it as the Google Maps for the brain. And in the big picture, all these trends points to one major shift, which is neuroscience. Neuroscience is moving from observation to intervention to enhancement. We are not just studying the brain anymore. We are starting to repair, reprogram, and even augment it. And we are entering the decade of the brain again. But this time we have the tools to mean it. And this reflects that earlier decades promised breakthroughs. Now, with AI plus genomics plus computing power, those breakthroughs are actually happening. And the brain defines who we are, yet we still understand only a small part of it. And I will leave you with this thought: if we fully understand the brain, will we redefine what it means to be a human? Thank you, and feel free to reach out for any questions.
Thank you, Jhansi, for that insightful lecture. Our next lecture will be delivered by Gorki Guha, who comes from a background in zoology with a special interest in neuroscience. Her lecture focuses on foundations of neuroscience, neural systems, disorders, and emerging insights. Over to you, Gorki.
Hello everyone. I'm Gorki Guha, and today I'll be talking about the foundations of neuroscience, encompassing neural systems, disorders, and emerging insights in the same. So, before we dive deeper, let us look quickly at the roadmap for today's presentation. We begin with the fundamental blocks of the nervous system, neurons and glial cells. Neurons are specialized cells responsible for transmitting electrical signals, chemical signals throughout the body. Glial cells are thought to be merely supporting cells. They were actually thought to be merely supporting cells, but now they are recognized as active participants in the brain communication, protection, and repair. Next, we will move into neural communication. This section will explain how neurons generate electrical impulses called action potentials and how they communicate across synapses using neurotransmitters. This is essentially the language of the nervous system. After that, we will examine the nervous system's organization. Specifically, we'll try to distinguish between the central nervous system and the peripheral nervous system. We will then explore the major neural systems involved in sensory processing, motor control, memory, emotion, and cognition. Following this, we will be discussing neurological disorders. We'll understand how disruptions in the neural circuit contribute to diseases such as Alzheimer's, Parkinson's, epilepsy, stroke, and multiple other neurodegenerative conditions. So, this includes now, finally, we will be concluding with emerging insights and future directions, which will include exciting fields such as neuroplasticity, brain computer interface, artificial intelligence in neuroscience, personalized neurology, and regenerative medicine. So, overall, this presentation moves from the cellular level all the way to the future neuroscience innovations. Now, we arrive at the most fundamental functional unit of the nervous system, the neuron. Neurons are specialized excitable cells designed for rapid communication. Unlike most cells in the body, neurons are uniquely adapted to transmit information over long distances using electrical and chemical signals. So, let us break down the major parts of a neuron. Dendrites. Dendrites are branching extensions that receive incoming signals from other neurons. You can think of dendrites as the antennae of the neuron. They collect information and carry it towards the cell body. Cell body, also known as soma. The soma contains the nucleus and essential cellular machinery. This is the metabolic center of the neuron where proteins are synthesized and cellular activities are regulated. Then comes the axon. The axon is a long projection responsible for transmitting electrical impulses away from the cell body. Some axons can extend over a meter in length, especially those connecting the spinal cord to the limbs. Then we come to myelin sheath. Many axons are wrapped in a fatty insulating layer called the myelin. Myelin greatly increases the speed of impulse conduction. Without myelin, neural signaling becomes slower and less efficient. Diseases such as multiple sclerosis occurs when this myelin is damaged. Then we have nodes of Ranvier. These are small gaps in the myelin sheath. Electrical impulses jump from one node to another in a process called saltatory conduction, allowing extremely rapid signal transmission. Axon terminal and synapse. At the end of the axon are axon terminals. They release neurotransmitters into the synapse, which is actually the microscopic gap between neurons. This is where electrical signals are converted into chemical signals for communication. Now, there are also different types of neurons, like multipolar neurons, which are commonly found in motor pathways, bipolar neurons associated with sensory systems, unipolar neurons, which are common in sensory neurons. Together, these structures allow neurons to detect, process, and transmit information with a remarkable speed and precision. While neurons receive most of the attention, glial cells are equally essential for nervous system's function. In fact, glial cells outnumber neurons in many regions of the brain. For many years, scientists believed glia only provide structural support. Modern neuroscience has completely changed that view. We now know glial cells actively participate in neural communication, immune defense, nutrient transport, synapse regulation, and tissue repair. Let us look at the major types shown here. Firstly, we have astrocytes. They maintain the chemical environment around the neurons. They regulate neurotransmitter levels, provide nutrients, and help form the blood brain barrier. The blood brain barrier protects the brain from harmful toxins and pathogens. Then we have oligodendrocytes, also known as Schwann cells. They will produce myelin in the central nervous system. One oligodendrocyte can myelinate multiple axons simultaneously. Now, Schwann cells will also perform a similar kind of role like oligodendrocytes. That is why I specifically said that interchangeably I am mentioning oligodendrocytes and Schwann cells. But the only difference between them is, unlike oligodendrocytes, each Schwann cell typically myelinates only one axon segment. Then we have microglia. Microglia are the immune cells of the brain. They remove damaged cells, pathogens, debris through phagocytosis. During injury or infection, microglia become highly active. Then we have ependymal cells. These cells line the ventricle of the brain and help produce and circulate CSF or cerebrospinal fluid. CSF cushions the brain and spinal cord while also transporting nutrients. So, although glial cells were once overlooked, they are now recognized as crucial partners in neural health, signaling, and protection. Now, let us explore how neurons actually communicate. Neural communication depends on two major processes. One, electrical signaling within neurons. Two, chemical signaling between neurons. Firstly, let's look at resting potential. At rest, neurons maintain a voltage difference across their membrane called the resting membrane potential. This is around minus 70 millivolts. The inside of the neuron will be negatively charged compared to the outside. This difference is maintained by ion channels and the sodium potassium pump. Then there will be a depolarization step and that happens when a stimulus reaches the threshold level and sodium channels open up rapidly. Sodium ions rush into the neuron. This causes depolarization where the membrane potential becomes more positive. Then we have the action potential. What is it? It's actually this rapid electrical change which creates this action potential, which travels down this axon like a wave. Importantly, action potentials follow the all or none principle. That is, either the threshold is reached and a full signal occurs or no signal occurs at all. Then we have repolarization, where the potassium channels will open up and potassium ions start moving out of the neurons. This restores the negative membrane potential. Now let's come to synaptic transmission. When the action potential reaches the terminal of the axons, neurotransmitters will be released in the synaptic cleft. These neurotransmitters bind to the receptors on the next neuron. Different neurotransmitters will produce different effects, like dopamine for reward and movement, serotonin for mood regulation, acetylcholine for muscle activation, GABA or gamma aminobutyric acid for inhibitory signals, glutamate for excitatory signaling. Thus, neural communication forms the basis of every thought, movement, memory, and emotion in the human body. Now, to understand neuroscience fully, we must understand how the nervous system is organized. The nervous system is broadly divided into two parts: the central nervous system and the peripheral nervous system. The central nervous system includes the brain, the spinal cord. They act as the command center of the body. The brain processes sensory information, generates thoughts, controls emotions, and coordinates behavior. The spinal cord acts as the major communication highway between the brain and the body. It additionally controls reflex actions. Then we have the peripheral nervous system. It will include all the nerves outside the central nervous system. It will connect the brain and the spinal cord to the muscles, organs, and sensory receptors. PNS further on is divided into the somatic nervous system and the autonomic nervous system. Somatic nervous system will control all of our voluntary actions like walking, writing, speaking, and skeletal muscle movement. Autonomic nervous system, on the other hand, regulates involuntary functions like blood pressure, gland activity, respiration, digestion, heart rate. Further on, autonomic nervous system is divided into two parts, sympathetic nervous system and parasympathetic nervous system. Sympathetic nervous system is also known as the flight or fight system. It prepares the body for stress by increasing the heart rate and energy availability. Parasympathetic nervous system, on the other hand, is known as the rest and digest system. It promotes relaxation, digestion, and recovery. Together, these systems maintain the internal balance or homeostasis. Now, let us examine the major neural systems responsible for sensation and movement. Sensory systems. Sensory systems will detect information from the environment. Specialized receptors convert external stimuli into electrical signals. These signals travel through sensory pathways to the brain, like vision, hearing, taste, smell, touch, pain, temperature. Different regions of the cerebral cortex will process different sensory inputs, like the occipital lobe is going to process vision, the temporal lobe processes hearing, the parietal lobe processes touch, spatial awareness. Now, the motor systems will be controlling movement. They will send signals through descending pathways to skeletal muscles. The cerebellum is going to control the balance, posture, and fine motor control. The basal ganglia regulates movement, initiation, and smooth execution. Now, damage to these systems can result in movement disorders like tremor, rigidity, and paralysis. Now, here we can also see that there is a linking between sensory input and motor input. This integration allows rapid responses to environmental changes. Suppose touching a hot object immediately triggers withdrawal reflexes. Then, visual information helps coordinate movement and balance. Thus, sensory and motor systems will work together continuously and help us interact with the world. Now, beyond sensation and movement, brain also supports higher mental function. Firstly, let's talk about hippocampus. Hippocampus will be essential for memory formation. It will convert short-term memories into long-term memories. Damage to the hippocampus will impair learning and memory retention. Then we have amygdala. Amygdala processes emotions, especially fear and emotional memory. It plays a major role in emotional reactions and survival behaviors. Hypothalamus. Hypothalamus maintains homeostasis. It regulates hunger, thirst, temperature, hormones, sleep, and anti stress responses. Prefrontal cortex. It is associated with decision making, planning, attention, personality, and executive control. This region is critical for rational thinking and complex behavior. Then we move to the limbic system integration. Limbic system integrates emotion, memory, and motivation. This explains why emotions strongly influence learning and decision making. Modern neuroscience increasingly recognizes that cognition and emotion are deeply interconnected rather than separate processes. The nervous system constantly monitors internal and external conditions. It then generates coordinated response to maintain stability. Now, one of the pathways commonly seen here will be the reward pathways. The reward system involves the dopamine pathways. These pathways reinforce behaviors associated with pleasure, motivation, and learning. However, overstimulation of reward circuits can contribute to addiction. Then, stress response. The hypothalamus activates the stress response through the hypothalamus pituitary axis, also known as the HPA axis. Stress hormones such as cortisol prepare the body to respond to challenges. Chronic stress, however, can negatively affect memory, immunity, and emotional health. Then, brain body integration. The brain communicates with the organs throughout the body constantly. For example, the nervous system regulates heart rate, controls breathing, maintains blood pressure, coordinates digestion, and controls body temperature. Feedback systems allow constant adjustments. For example, balance depends on signals from the inner ears, eyes, and muscles. Body temperature regulation depends on sensory inputs and hypothalamic control. Thus, the nervous system functions as a dynamic regulatory network that keeps the body alive, adaptive, and responsive. Despite its incredible complexity, the nervous system is vulnerable to disease and injury. Neurological disorders arise when neurons, synapses, or neural circuits become damaged or dysfunctional. Firstly, talking about Alzheimer's disease, it's a progressive neurodegenerative disorder. It is associated with memory loss, cognitive decline, accumulation of amyloid plaques, and tau tangles in the brain. As neurons degenerate, communication between brain regions deteriorates. Then Parkinson's disease. Parkinson's primarily affects movements. It results from degeneration of the dopamine producing neurons in the substantia nigra. Symptoms include tremors, muscle rigidity, slowed movements, and postural instability. Then we have stroke. It occurs when the blood supply to part of the brain is interrupted. Neurons deprived of oxygen can come under hypoxic conditions and can die within minutes. Depending on the affected region, stroke may impair speech, movement, memory, or sensation. Epilepsy. Epilepsy involves abnormal electrical activity in the brain leading to seizures. These seizures can vary from brief lapses in awareness to severe convulsions. Then multiple sclerosis, it's an autoimmune disorder and it damages the myelin in the CNS. As neural transmission weakens, symptoms such as weakness, vision problems, and coordination issues emerge up. Understanding these disorders will help neuroscientists develop better treatments and therapies. One of the most revolutionary discoveries in the field of neuroscience is neuroplasticity. The brain is not fixed. Instead, neural circuits can reorganize and adapt throughout life. This means learning, rehabilitation, and recovery are biologically possible. Then we have BCI, brain computer interfaces. They will allow direct communication between brain and external devices. These technologies may help restore movement in paralysis and improve communication for patients with severe neurological conditions. Then we have AI in neuroscience. Artificial intelligence is now helping scientists analyze massive neural data sets. Machine learning can identify patterns linked to disease such as Alzheimer's and epilepsy. AI assisted neuroimaging is improving diagnosis and precision medicine. Neuroregeneration. Researchers are exploring stem cells and regenerative therapies to repair damaged neurons. This could transform treatment for spinal cord injuries and neurodegenerative disorders. Precision neurology. Future medicine may become highly personalized. Genetic profiling and advanced imaging may allow treatments tailored specifically to individual patients. These innovations demonstrate that neuroscience is entering an era of extraordinary transformation. While neuroscience offers incredible possibilities, it also raises major ethical and societal questions. Firstly, as neurotechnology advances, protecting brain data becomes increasingly important. If thoughts or neural patterns can be decoded, issues of privacy and consent become critical. Then, artificial intelligence may influence diagnosis and treatments. However, excessive dependence on algorithms again raises questions regarding biasness, transparency, and accountability. Future technologies may enhance memory, intelligence, or attention, but this raises another ethical question: who gets the access? Could enhancement increase inequality? And where do we draw the line between therapy and enhancement? Brain computer interfaces. Direct brain device communication could transform medicine. Yes, it does, but it also creates concerns regarding cybersecurity and misuse. Then, advanced neuroscience treatments may be expensive. Ensuring equal access across population thus remains a major challenge. Therefore, neuroscience must progress alongside ethical responsibility. As we approach the conclusion, I would like to emphasize on the point that human brain is often described as the most complex object in the known universe. It is capable of conscious thought, creativity, emotion, learning, adaptation, and self awareness. Neuroscience itself bridges multiple disciplines like biology, medicine, psychology, engineering, computer science, and artificial intelligence. From understanding single neurons to decoding entire neural networks, neuroscience continues to reshape our understanding of what it means to be human. The future may include more effective treatments for neurological disorders, brain controlled prosthetics, personalized neural therapies, and deeper understanding of consciousness itself. Yet, with these advances comes the responsibility to use neuroscience ethically and wisely. Ultimately, studying the brain is not only about understanding neurons, it is about understanding ourselves. Thank you.
Thank you, Gorky, for that well structured and thoughtful lecture. Our next lecture will be delivered by Sanampreet Kaur, who comes from a background in human genetics, and she has been actively working on and educating people in neuroscience. Her lecture focuses on BDNF and depression, a neuroplasticity framework for mood disorders. Sanampreet, please go ahead.
Hello, everyone. Today I'll be talking about depression from a neuroplasticity perspective, with a focus on BDNF or brain derived neurotrophic factors, one of the key molecules involved in that. Before we move into BDNF, let's first discuss the research framework that has shaped the depression research for decades. And I'm talking about the monoamine hypothesis. So, monoamine hypothesis basically states that depression results from the reduced level of important neurotransmitters, serotonin, norepinephrine, dopamine, and each of these neurotransmitters play a distinct role in the brain. Serotonin is responsible for regulation of mood, sleep, appetite, norepinephrine for arousal, attention, and for stress response, while dopamine is important for reward, motivation, and reinforcement learning. Now, pharmacologically, this model seems to be very good because there are drugs like antidepressants, like SSRIs, SNRIs. They increase the availability of these neurotransmitters in the synaptic cleft. One of the most commonly prescribed drugs, SSRIs, block the uptake of serotonin from the synapse by blocking SERT or serotonin transporter, and thus it rapidly increases the level of serotonin at the synapse, often within the hours. But there is a catch. While neurotransmitter levels rise quickly at the synapses, but the clinical improvement often take weeks. And this temporal gap, suggests that monoamines are not the final step in the recovery, but they're more like the triggers for the slower and more downstream changes that are happening. And this has been supported by the neuroimaging studies that consistently reveal reduced hippocampal volume and decreased connectivity in the prefrontal cortex. And these are not just chemical imbalances, these are structural changes. And then there is another problem. Not all the patients respond to monoaminergic treatments, and even when they do, remission is often partial. So, monoamines may influence the mood, but they do not fully explain the structural, functional, and temporal complexity of depression. Now, this brings us to a deeper question. What molecular systems actually drive these long term changes in the brain structure and connectivity? And this is where BDNF enters the story. BDNF, or brain derived neurotrophic factor, is part of the neurotrophin family. There are other neurotrophins also. Basically, this family is comprised of a group of proteins that regulate the neuronal survival, growth, and plasticity. And BDNF is one of the most important ones. It is highly expressed in brain areas which are important for mood and for cognition, particularly the hippocampus and the prefrontal cortex. At the cellular level, BDNF acts as a regulator for both the stability and the change of the neural circuits. It supports the neuronal survival, protecting them from degeneration. It also drives the synaptic growth, promoting dendritic branching and the formation of new synapses. And finally, it is also essential for the circuit formation and circuit refinement, which involves strengthening some synapses while allowing the others to weaken to make the circuits more efficient. In other words, BDNF actively shapes the brain's wiring. Now, what's really interesting about BDNF is that it operates like a two-phase system. BDNF is first synthesized as a precursor protein called pro-BDNF, which can be later cleaved into mature form or mature BDNF. And both are producing the opposite biological effects. Now, mature BDNF preferentially binds to the tyrosine kinase B receptor family, and it activates several intracellular signaling pathways, and together these pathways are actually leading to synaptic strengthening, neurite outgrowth, and enhanced neuroplasticity via enhancing the long-term potentiation. In contrast, the pro-BDNF preferentially binds to the P75 receptor, and which is often activated alongside the co-receptors like sortilin. And these receptors are activating a very different set of intracellular pathways, which are associated with apoptosis and cell death, dendritic spine retraction, and even synaptic weakening or long-term depression. Under normal conditions, your brain carefully regulates the balance between pro-BDNF and mature BDNF to maintain healthy plasticity in your brain. BDNF is actually a key mediator of long-term potentiation or LTP. During LTP, there is repeated synaptic activity that leads to sustained increase in synaptic strength. At active synapses, neuronal activity triggers the activity-dependent release of BDNF, and this BDNF then binds to its respective receptors, the tyrosine kinase B receptors, which enhances the synaptic efficiency. And these processes convert the short-term activity into long-term and lasting synaptic strengthening, and it also regulates the maintenance of dendritic spines. And these spines are actually not static, they're constantly being remodeled. New spines are formed in response to experience, and existing synapses can grow, stabilize, or even retract. And BDNF supports the spine maturation and stability, ensuring that important connections are preserved over time. This is essential not only for learning and memory, but also for emotional regulation, because experiences like stress, reward, social interaction, all depend on your brain's ability to adapt its circuitry. When BDNF signaling is intact, synapses remain flexible, circuits can reorganize, and the brain can adapt to changing environments. But when BDNF levels are reduced, LTP would be impaired, dendritic spines are lost, and synaptic connections weaken over time. And the result is that the brain becomes less flexible, less adaptive, and less capable of change, something that we see in depression. But chronic stress offsets this balance, which is mediated by BDNF. Chronic stress is one of the most consistent biological risk factors for the development of depression, and its impact is mediated by the hypothalamic pituitary adrenal axis or HPA axis. Under prolonged stress, your HPA axis remains persistently activated, which results in chronically increased levels of cortisol. Now, this cortisol readily crosses the blood-brain barrier and it reaches in the important brain regions like hippocampus, prefrontal cortex, which are expressing heavily the receptors for cortisol. And in short term, this is actually beneficial for you because cortisol helps you to adapt to the environmental stressors. But with chronic activation, it actually begins to disrupt the cellular functions. One of the most critical effects is the suppression of BDNF signaling. This is because of the reduced expression of transcription factors like CREB, or even due to epigenetic repression. So because of this decline in the BDNF availability, your neuron's ability to maintain and remodel synaptic connections also changes and reduces. At the same time, stress also shifts the balance between the levels of mature BDNF and pro-BDNF. There is a relative increase in the pro-BDNF signaling, and this pushes the entire neuronal system towards synaptic weakening, dendritic spine retraction, and apoptotic signaling. This is not a sudden collapse. It is a slow, progressive decrease in the neuroplasticity, and over time, your brain becomes less and less capable of adapting to new experiences or recovering from the negative states. The question that arises here is that do these microscopic changes actually scale up to the level of brain structure? And the answer is yes, and we can directly see it in neuroimaging and the postmortem studies. One of the most consistent findings is reduction in the hippocampal volume. Now, hippocampus, we know that is expressing high density of cortisol receptors, which makes it highly vulnerable to chronic stress. And when BDNF levels decline, neurogenesis and synaptic density also gets reduced in hippocampus. And over time, this leads to measurable loss of volume in hippocampus, which correlates with the memory and stress regulation impairments. In prefrontal cortex also, we see similar structural changes, especially in the dorsolateral and the medial prefrontal cortex. Reduced BDNF contributes to cortical thinning, and functionally, this weakens the brain's top-down control of emotions, making it more and more difficult to regulate negative emotions and negative emotional states. And we see similar changes across multiple other brain regions as well. Connecting molecular and behavioral changes, BDNF operates at the level of individual circuits. And these circuit disruptions manifest as the symptoms that we recognize in depression, like low mood, anhedonia, or even cognitive deficits that are linked to weakened prefrontal, hippocampal, and other circuits that we're going to discuss now. Let's start with the first circuit, the prefrontal cortex amygdala circuit. Amygdala acts as an early warning system which detects the emotionally salient stimuli, especially those that are linked with some kind of threat or negative effect, and it rapidly initiates the emotional and stress responses. On the other hand, the prefrontal cortex provides the top-down regulation, and it basically evaluates and applies the cognitive control and modulates the intensity and the appropriateness of the emotional responses. So under healthy conditions, there is a dynamic balance between the activity of amygdala and the prefrontal cortex. The amygdala generates the emotional signal, the prefrontal cortex refines and regulates them, and BDNF is necessary for maintaining this balance. But under chronic stress, the BDNF levels decline and this balance begins to shift. Prefrontal cortex loses its regulatory grip over amygdala and amygdala becomes overactive. And this essentially manifests as heightened negative affect, increased stress sensitivity, and impaired emotional regulation. Behaviorally, we observe persistent low mood, exaggerated stress responses, difficulty controlling emotions, and it's just not a chemical issue, it is a failure of the circuit level control. Now we move on to memory and stress circuit involving hippocampus. In a healthy brain, hippocampus plays an important role in regulating the body's stress response. It interacts with the HPA axis, and whenever a stressor appears, there is release of cortisol, which then feedbacks onto the brain. It binds to the receptors in the hippocampus, and under normal conditions, the hippocampus acts like a brake. It detects the elevated cortisol levels and sends the signal back to shut down the stress response, which restores the balance. But under chronic stress, this system begins to break down because there is sustained level of cortisol, because of which there is suppression of BDNF expression. And as BDNF levels decline, the hippocampus undergoes structural atrophy, and its ability to regulate the HPA axis becomes weaker and weaker. And this creates a reinforcing feedback loop. Stress increases cortisol, cortisol then suppresses the BDNF, Reduced BDNF weakens the hippocampus. A weakened hippocampus fails to inhibit the HPA axis, which further leads to increase in the cortisol levels, and this maladaptive loop goes on that sustains and amplifies the depressive state. Behaviorally, it manifests as the persistent stress sensitivity and difficulty in recovering from negative experiences and impairments in memory and contextual processing. Finally, there is involvement of the reward circuitry. The experience of reward and motivation is generated by the mesolimbic dopaminergic system, the pathway which is connecting the ventral tegmental area to the nucleus accumbens. Under normal conditions, the dopaminergic neurons in the ventral tegmental area project to the nucleus accumbens as well as to the prefrontal cortex. Whenever we encounter something that could be meaningful to us or could feel as rewarding, these neurons increase the dopamine release, and which reinforces the behavior and drives the goal-directed actions. Nucleus accumbens acts as an integration hub because it receives dopaminergic input as well as the glutaminergic input from the cortical and the limbic areas, both of them, allowing the brain to assign values to experiences. But when BDNF signaling is disrupted, it leads to impaired synaptic plasticity in the nucleus accumbens. It also leads to the altered dopaminergic transmission dynamics, which basically manifests as the reduced ability to strengthen the circuits in response to rewarding stimuli. So in other words, the reward system becomes blunted. The stimuli which were once meaningful to us, they fail to generate enough dopaminergic response. And motivation declines, actions are no longer linked to rewards. Clinically, this is experienced as something we call anhedonia, or a reduced capacity to feel pleasure, interest, or even drive. Now let's get back to the serotonin and try to solve one of the most enduring puzzles in the depression treatment, which is the therapeutic delay which is seen with the antidepressants. When a patient takes an SSRI, the serotonin levels in the synaptic cleft increase within the hours due to inhibition of serotonin transporter. But the clinical improvement takes weeks. If depression were simply because of low serotonin, the symptom relief would also happen as quickly as the increase in the serotonin levels. But it doesn't. Because at the molecular level, the increased serotonin signaling activates the intracellular pathways like CAM protein kinase A pathway and the transcription factors like CREB. CREB in turn increases the expression of genes involved in neuroplasticity, especially the BDNF. With repeated treatments, BDNF levels rise and they activate the TRKB receptors, and the further intracellular signaling happens. There is synaptic strengthening, dendritic branching takes place, synaptic spine density increases, and the neural circuits begin to reorganize and restabilize. But these are not the immediate chemical shifts. They are structural and functional changes that are happening in the brain, and they actually take time. Now we have solved the puzzle. The delay in the clinical response is actually because of the time it takes to stabilize the neuroplasticity, to restore the neuroplasticity in the brain. But the most interesting part is that there is actually an antidepressant which has bypassed this temporal delay. I'm talking about ketamine. Unlike traditional antidepressants, ketamine produces rapid antidepressant effects, often within the hours, even in treatment-resistant patients. Ketamine actually works primarily as an NMDA receptor antagonist on the GABAergic interneurons in the prefrontal cortex. So basically, it blocks the NMDA receptors on the inhibitory neurons, and it reduces the inhibition in the prefrontal cortex, releasing a brake. So there is a rapid surge in the glutamate levels in the pyramidal neurons, and this glutamate then activates the AMPA receptors, NMDA receptors, and it leads to what we call the long-term potentiation, which results in the enhanced neuroplastic changes in the brain. So unlike SSRIs, which depend on the gradual gene transcription and remodeling, ketamine actually induces the activity-dependent plasticity almost immediately. And this rapid restoration of connectivity is also reflected clinically There is quick improvement in the mood, there is reduction in anhedonia, and there is even decrease in the suicidal ideation within hours. From a neuroplasticity perspective, this offers a very strong evidence that the core issue in depression is not just the altered neurotransmitter levels, but a disruption in the brain's ability to form and maintain functional connections. Now, restoring neuroplasticity in depression is not just limited to medication. There are a range of behavioral neuromodulatory interventions that converge on the same biological pathways, that is the BDNF related neuroplasticity pathways. Let's first start with the physical exercise. Regular aerobic exercise is one of the most reliable natural stimulators of BDNF expression, especially in the hippocampus. Exercise increases the neuronal activity, which leads to the calcium influx. There is activation of signaling pathways, including CAM, kinase 2 pathways, and the Krebs transcription factors, which lead to the release of BDNF. It also improves the cerebral blood flow, providing the metabolic support and promoting the adult neurogenesis in the dentate gyrus. Functionally, this translates as improved mood, cognition, and stress resilience. Next, we have the cognitive behavioral therapy or CBT. And CBT actually operates at the psychological level, but the effects are also biologically measurable. By repeatedly engaging in a new pattern of thought and behavior, there is activation of specific neural circuits, which drives the experience-dependent synaptic plasticity. Over time, the maladaptive circuits weaken, while the adaptive ones are strengthened. And this process is also supported by the BDNF-dependent mechanisms. Finally, we have the transcranial magnetic stimulation or TMS. Now, TMS is a non-invasive technique that uses magnetic fields to stimulate the targeted brain regions, particularly the prefrontal cortex, which are underactive in depression. At the cellular level, TMS modulates the neuronal excitability. It induces long-term potentiation like plasticity, and it also increases the BDNF expression in the stimulated circuits. Now, all these interventions, exercise, psychotherapy, transcranial magnetic stimulation, they're all converging on the shared biological endpoint, which is BDNF, which further reinforces the central idea of this talk, which is that depression is about the brain's lost ability to adapt, to reorganize, and to change. Now, this brings us to an important question. Why do some individuals develop depression under stress while others remain relatively resilient? Part of that answer lies in the genetic variation, particularly within the BDNF gene. One of the most well-studied variants is Val66 MET polymorphism. This basically involves change in one nucleotide, which leads to change in the amino acid from valine to methionine at codon number 66 in the pro domain of BDNF. On first glance, this appears like a very small change, but functionally, there are significant consequences. This polymorphism specifically affects the activity-dependent secretion of BDNF. So under normal situation, neuronal activity triggers the release of BDNF at the synapses, which supports the synaptic strengthening and the plasticity. But individuals who are carrying the MET allele, in those individuals, BDNF is missorted within the cells and its regulated release is reduced. So even when the neurons are active, the amount of BDNF which reaches the synapses is much less than a normal individual's. So this polymorphism does not directly cause depression, but it influences the vulnerability. It makes the carriers for this polymorphism less able to mount the plasticity-based responses, making them more susceptible to the effects of stress. Another factor which influences a person's susceptibility to develop depression is epigenetics. Epigenetics basically include the mechanisms that regulate gene expression without actually changing the DNA sequence. BDNF gene is especially sensitive to these epigenetic changes. One of the primary mechanisms which drives these changes is activation of the neurodegeneration pathway. there is increased methylation at specific BDNF gene promoters, which reduces the binding of transcription factors like REB, and which further reduces the BDNF transcription. Another mechanism involves histone modifications. DNA is actually wrapped around histone proteins, and the chemical changes to these proteins determine how accessible a gene will be. Histone acetylation loosens the chromatin and promotes gene expression, while histone deacetylation makes chromatin more compact and suppresses the transcription. In depression, there is a shift towards more repressive chromatin states, which further reduces the BDNF expression. Now, epigenetics provides a direct bridge between experience and biology, and stressful experiences are not just being processed at a psychological level. Epigenetics proves that these are being translated into molecular levels, which alter how genes are expressed. However, there are several important limitations of this BDNF model that we need to consider. First, there is inconsistent clinical evidence. There are many studies which report reduction in the BDNF levels in depression, which increases with treatment, but the findings are not always consistent, which may be because of variability in the methods that measure the peripheral BDNF levels. Second, there is the problem of regional specificity. Now, BDNF does not act the same way across all the brain regions. In areas like hippocampus and prefrontal cortex, as we have already discussed, it supports adaptive plasticity. But in regions like nucleus accumbens and under certain conditions, BDNF can actually contribute to maladaptive changes. So the levels of BDNF would be normal in these regions, and they would be contributing to maladaptive behavioral patterns. Third, there is this problem of therapeutic variability. Not all the treatments would work through BDNF levels, BDNF pathways. Some patients respond to interventions which are not strongly associated with the BDNF levels. This is probably because of involvement of other interacting systems which contribute to the depression pathophysiology, like inflammation, alteration in the glutaminergic signaling, and endocrine regulation. And fourth, there is this question of causality versus correlation. It's hard to say whether reduction in the BDNF levels is a cause of depression or it is because of the consequence of chronic stress and altered neural activity. And finally, there is this risk of oversimplification. Just as the monoamine hypothesis once reduced depression to a chemical imbalance, there is a similar risk of reducing it to the BDNF deficiency model. But depression is far more complex for that. So rather than viewing depression as a chemical imbalance or simply as a BDNF deficiency, it is much more accurate to understand it as a multi-level systems disorder, where molecules interact with genes, synapses reshape circuits, circuits generate behavior, and experience continuously feedbacks onto biology. Within this framework, BDNF remains central because it connects these levels. So the question is no longer which single chemical is missing, but instead, how do multiple systems converge to shape the brain's capacity to adapt? And ultimately, recovery from depression may depend not only on correcting one imbalance of brain chemical, but on restoring the brain's ability to change, reorganize, and respond to experience. The final takeaway is this: depression is not just a disorder of mood, it is a disorder of adaptability, and treatment at its core is the restoration of this change itself. Thank you.
Very well explained, Sanampreet. Thank you. We now move into the theme 4, mechanisms of neurodegeneration, disease pathways, models, and therapeutic strategies. This session will focus on understanding the underlying biological mechanisms of neurodegenerative diseases and how modern research is working toward addressing these challenges. Our first lecture in this session is again by Gorky Guha. Her lecture is exactly on mechanisms of neurodegeneration. Welcome, Gorky.
Hello everyone, I'm Gorky Guha and today I'll be talking about the mechanisms of neurodegeneration, which will encompass the disease pathways, models, and therapeutic strategies. We'll be specifically looking into the shared as well as the distinct mechanisms of Alzheimer's, Parkinson's, amyotrophic lateral sclerosis, also known as ALS, and Huntington's disease. We'll explore how neurons, the cells that make us think, move, and remember, slowly lose their function in diseases like Alzheimer's, Parkinson's, etc. So, in today's presentation, I would like to connect the molecular mechanisms to real world challenges and emerging therapies in this area. By the end, I hope you'll see how understanding these pathways isn't just about science, it's about hope for millions of lives. So, here's our roadmap for today. We'll start with an introduction to neurodegenerative diseases, what they are and why they matter. Then we'll dive into common and disease specific pathways, exploring how different diseases share overlapping mechanisms. Then we'll look at the experimental models, both in vitro and in vivo, which will help us study these processes. After that, we'll discuss therapeutic strategies from symptomatic treatments to cutting edge disease modifying approaches. Finally, we'll end with challenges and future directions, where I'll show you how precision medicine and AI are reshaping this field. Think of this as a journey from understanding how neurons die to discovering how we can help them live and thrive. Neurodegenerative diseases are conditions where neurons progressively lose their structure, function, and unlike other cells, neurons rarely regenerate. They primarily affect older adults, but early onset forms exist too. So let's look at a few examples. Alzheimer's disease, AD, affects memory and cognition. Parkinson's impacts motor control. Amyotrophic lateral sclerosis destroys motor neurons, and Huntington's, caused by CAG repeat expansion, leads to movement and psychiatric symptoms. Globally, more than 50 million people are affected, with over 10 million deaths annually, and an economic burden exceeding more than $1 trillion. So, neurodegeneration isn't just a biological problem, it's a global health crisis and a societal challenge. When we look across these diseases, we find eight recurring hallmarks, which are actually the molecular fingerprints of degeneration. They include protein aggregation. Misfolded proteins will be forming toxic clumps, synaptic dysfunction, where a communication between the neurons break down, neuronal cell death, where there's an irreversible loss of brain cells, endoplasmic reticulum proteostasis impairment, where there's a failure of cellular protein quality control, cytoskeletal abnormalities, where there's a disruption in the axonal transport, DNA and RNA damage leading to genetic instability, neuroinflammation, where there's a chronic activation of microglia and astrocytes, and mitochondrial dysfunction, where there's an energy failure and oxidative stress. These hallmarks overlap across the diseases, showing that neurodegeneration is not one disorder but a spectrum of interconnected failures. One of the most universal mechanisms in these diseases will be protein misfolding. Proteins are meant to fold into precise shapes to function correctly, but when folded wrong, they form toxic oligomers and aggregates. In Alzheimer's, we get to see beta amyloid fibrils. Sorry for the spelling mistake here. And then there will be hyperphosphorylated tau tangles, and both of them will accumulate. In Parkinson's, there will be alpha synuclein protein that forms Lewy bodies, and then we have TDP43 in ALS, which upon being aggregate, which actually is going to disrupt RNA metabolism when it starts aggregating. Now, these aggregates, whether it's in Alzheimer's, whether it's in ALS, whether it's in Parkinson's, they will start spreading from cell to cell like a prion. It's like a prion like propagation, turning thus neurodegeneration into a contagious process within the brain. It's like a molecular wildfire. Once ignited, it spreads relentlessly. So now we look into this disruptive feedback loop. Sorry, disruptive feedback loop that drives all the neurodegenerative diseases. It's like a vicious loop. So it starts with oxidative stress, where the reactive oxygen species damage the mitochondria. Damaged mitochondria will leak out more of ROS, worsening the stress. Meanwhile, neuroinflammation kicks in, where microglia releases cytokines like TNF alpha, interleukin 1 beta, amplifying the damage. Then we have mitochondrial dysfunction, which leads to energy failure, calcium overload, and apoptosis. Protein aggregation and autophagy dysfunctions will further clog the system, and eventually blood brain barrier breakdown and genetic and environmental factors also kick in, and we get a self perpetuating cycle like a storm of cellular chaos that keeps feeding itself. So now let's compare the major diseases side by side. First, we will have Alzheimer's, where A, beta, and tau. A beta in the sense that amyloid beta fibrils and tau will accumulate in the hippocampus and cortex, leading to memory loss. And these proteins will be actually forming plaques and tangles. In Parkinson's, we will see alpha synuclein protein accumulation, which will be forming the Lewy bodies, and usually it's the substantia nigra which is affected. Here there is a loss of the dopaminergic neurons, and we also get to see some motor symptoms getting developed here. Then we have amyotrophic lateral sclerosis, where the primary proteins involved will be superoxide dismutase 1, SOD1, and TDP43. They are usually accumulating in the motor cortex and spinal cord, and they lead to motor neuron death and muscle atrophy. Another neurodegenerative disease which is not shown here is Huntington's. In Huntington, there is this mutant protein called Huntington, and it's mutated because there is an increase in the number of CAG repeats in the protein. This is going to lead to psychiatric symptoms eventually. So each disease has its own unique molecular triggers, but all of them converge on shared outcomes, which is energy failure, inflammation, and neuronal death. To study these mechanisms, we rely on experimental models that recreate the disease pathology. In vitro models include cell lines, primary neurons, induced pluripotent stem cells derived neurons, 3D organoids, and brain on a chip system, which is ideal for controlled high throughput studies. In vivo models will include transgenic rodents, non-human primates, non-mammalian species like Drosophila, C. elegans, and zebrafish. They'll be perfect for studying systematic effects and behavior. Together, these systems allow us to test hypotheses, screen drugs, and understand disease progression from molecules to whole organisms. In vitro systems are powerful because they are human relevant and scalable. We can grow iPSCs from patients to study disease in a dish. 3D organoids, on the other hand, mimic mini brains capturing complex interactions. Microfluidic chips simulate axonal transport and neural networks. These models are perfect for studying protein aggregation and cellular stress. So their advantages, we have a controlled environment, high throughput screening, and human relevance. But again, there is a pitfall to it. They lack full brain complexity. There is no vascular or no immune components involved here. So they are great for mechanistic insight, but fall a step back for whole system behavior analysis. So, animal models bring back the complexity of living systems. Rodents are the most common. Transgenic lines like APP or PS1 for Alzheimer's or alpha synuclein for Parkinson's replicate key features. toxin induced models like MPTP mimic dopaminergic neuron loss. Non mammalian models like flies, in this context, Drosophila, of course, then C. elegans, zebrafish, they are fast and cheap for genetic screens. Non-human primates are closest to humans but raise ethical and cost concerns again. So these models they help us study systemic effects, behavior and disease progression, but species differences in brain anatomy and lifespan still remain as a major challenge. So, therapies will fall into two categories in the context of neurodegenerative diseases. Firstly, we have symptom based treatments which will manage symptoms without altering disease progression, like choline esterases. They will be inhibiting. They'll act as inhibitors for Alzheimer's disease. Then we have levodopa for Parkinson's, riluzole for ALS. Then there are disease modifying therapies which aim to tackle the root causes. They'll be clearing the protein aggregates, reducing inflammation, restoring mitochondrial function, and using gene therapy for long term protection. So, the shift from symptom relief to disease modification marks a turning point from coping to curing. So, now let's talk about broader therapeutic approaches in neurodegeneration. So, scientists are exploring broader strategies which will tackle the overlapping hallmarks of neurodegeneration. We have neuroinflammation drugs like NLRP3 inhibitors and TREM2 modulators aim to calm overactive microglia and reduce harmful cytokine release. Then compounds like SIRT3 activators, mTOR inhibitors, restore energy balance and cellular cleanup. Using ASOs, antisense oligonucleotides, or CRISPRs to silence or correct mutant genes can also be done, for example, in case of Huntington or SOD1 and ALS. Then, using nanotechnology, we can have nanoparticles engineered which will cross the blood brain barrier and deliver drugs directly to the neurons. Then, there is also drug repurposing. Surprisingly, metabolic drugs like GLP-1 analogs used in diabetes now show promise in improving brain health. The key idea here is a multi target or combination therapy since neurodegeneration involves converging pathways. Inflammation, proteostasis, metabolism hitting just one target thus isn't enough. So the future lies in integrated approaches that tackle several mechanisms all at once. So, of course, translating all these discoveries into real treatment is far from easy. Now, let's see the roadblocks that we face on the path to effective therapies. Firstly, animal model limitations. Rodents and other models often fail to predict human outcomes. What works in mice will not always work in people. Then there is this blood brain barrier penetration. Many drugs simply can't reach the brain in effective concentrations. Then we have disease heterogeneity. Every patient is different. Their genetics, environment, lifestyle is all different, making one size fits all treatments ineffective. Then there's this case of late diagnosis, so that is by the time symptoms appear, pathology is already advanced. And then we have clinical trial failures. Poor target selection and lack of patient stratification leads to such disappointing results. So, after this, we have biomarker gaps. We desperately need better fluid and imaging biomarkers that will detect disease early and track their progression. So, while science is advancing, the transition into therapies is slowed by these barriers. The message here is clear. To succeed, we need smarter models, earlier detection, and personalized approaches. So, now after looking at all the challenges, let's turn towards the future. And it's not bleak at all. This is where the things get exciting. The next decade of neurodegeneration research is all about precision, integration, and innovation. Precision medicine. Imagine tailoring treatments to each patient's genetic profile and biomarker signature. Instead of this one size fits all drug, therapies will be matched to the individual using genetic testing, fluid biomarkers, and imaging to select the right patients at the right stage. This means earlier intervention and better outcomes. Advanced models. We are moving beyond simple cell cultures. Human iPSCs, derived neurons, 3D organoids, and multi-omics integration will allow us to recreate human brain pathology in the lab with unprecedented accuracy. These models will bridge the gap between animal studies and human trials. Convergent therapies. Since neurodegeneration involves overlapping hallmarks, inflammation, proteostasis, metabolism, the future thus lies in combination therapies. Instead of targeting just amyloid or tau, treatments will simultaneously address multiple pathways, breaking the vicious cycle we saw earlier. Early detection and prevention. This is crucial. By the time symptoms appear, much of the brain is already damaged. The future will focus on predictive biomarkers, which will be blood tests, CSF markers, advanced imaging to detect disease years before clinical symptoms start appearing. Prevention will become as important as treatment. Then we have this AI and machine learning aspect. So, artificial intelligence is revolutionizing drug discovery. Algorithms will scan massive data sets to identify new targets, design molecules, and even predict clinical trial outcomes. This will accelerate the pipeline and reduce failures. And speaking of pipelines, the 2025-2026 horizon already looks promising. We have oral small molecules in development, next generation antibodies with better safety profiles, and metabolic therapies linking brain health to systemic metabolism. So the message is clear. The future of neurodegeneration research is hopeful, multidimensional, and patient centered. We are moving from treating symptoms to preventing the disease, from isolated targets to integrated strategies, and from trial and error to precision medicine powered by AI. Thank you.
Thank you, Golgi, for that comprehensive overview. We now welcome Sohit K. Reddy from a bioinformatics and computational biology background. His lecture is on amyloid cascade pathway in Alzheimer's disease. Welcome, Sohit.
Yeah, am I audible?
Yes, you are.
Yeah, thank you. So, hi everyone, this is Sohit. Today's presentation focuses on one of the most intensively studied and therapeutically significant pathways in all of the neuroscience, which is the amyloid cascade pathway in Alzheimer's disease. So, here we'll trace the molecular events from a single misprocessed protein all the way to the neuronal death. And also, we'll try to examine the experimental models that let us study this process. And then we'll also have a look at the cutting edge therapies out there that have recently crossed the finish line into clinical use, some for the very first time in a 30-year history of Alzheimer's research. So let's begin. So before diving into the molecular neuroscience, I want to ground us in like why this matters at the population level. So as of 2019, 55 million people worldwide live with dementia, the majority of whom have the Alzheimer's disease. And by 2050, the number is projected to reach over 139 million, driven largely by aging population in Asia and also sub-Saharan Africa. And the economic toll is pretty much staggering with like 1.3 trillion US dollars annually in global cost. And in the United States alone, the care costs are projected to exceed 409 billion US dollars in 2026. So if you have a look at the statistics, Alzheimer's actually kills more people than breast cancer and prostate cancer combined, yet it has received far less research funding per death until very recently. And these numbers make it pretty much clear this is not just like a scientific problem, it's a global health emergency. So there are several competing theories of Alzheimer's neurodegeneration, tau-centric, neuroinflammation first, and metabolic dysfunction. But here in this lecture, we are focusing on amyloid cascade hypothesis for three compelling reasons. First, it has over 30 years of evidence since Hardy and Higgins first proposed in 1992. Second, and most critically, it has genetic validation. Every known mutation that causes early onset familial Alzheimer's disease in APP, PSEN1, and PSEN2, it increases amyloid beta production, or its amyloid beta 42 is to 40 ratio. So you basically cannot argue with genetics. And third, we have the FDA approvals of lecanemab in 2023 and donanemab in 2024, the first ever disease modifying therapies that directly target amyloid, and they work. So now let's have a look at this slide, APP processing, normal versus amyloidogenic. So basically, amyloid beta does not exist in a vacuum in simple sense. It is produced from a normal transmembrane protein called APP, which is called amyloid precursor protein. And under the very healthy conditions, it is cleaved by an enzyme called alpha secretase inside the amyloid beta region. And this produces a soluble neurofragment called SAPP alpha. And the key point here is that the alpha secretase cuts within the amyloid beta sequence so that no amyloid can form. So this is called the non-amyloidogenic pathway, like a normal pathway. However, in Alzheimer's disease, the balance shifts. So here BACE1, which is a beta secretase enzyme, cleaves APP, which is the amyloid precursor protein. It cleaves the at the end terminus of the alpha beta amyloid beta region, releasing a C99 fragment. And then also we have gamma secretase, complex containing penicillin 1 and penicillin 2, which then cleaves C99 in order to produce or release amyloid beta 40 or amyloid beta 42. So basically, mutations in PSEN1 or 2 skew this cut to produce more amyloid beta 42, like the longer, stickier, more aggregation prone form. And the four genetic risk factors are in the right panel over here, which are BACE1, PSEN1 or 2, APP, and APOE4. Are the four core drug targets of the entire Alzheimer's field so far. And this is basically the heart of the presentation. The cascade has 5 sequential steps, and it's very much important to understand that steps 1 through 3 can occur silently for like 15 to 20 years before any symptoms can appear. The step 1 is the amyloid beta overproduction or impaired clearance. Here is where the soluble amyloid beta 42 monomers begin to accumulate. And the second step is where these monomers oligomerize into small soluble assemblies, which are like actually the most synaptotoxic species, which impairs long-term potentiation, the cellular basis of memory, basically. And step 3 is where these oligomers aggregate further into fibrils, which is called the amyloid beta fibrils, that deposit as extracellular plaques, which then triggers microglial activation and neuroinflammation. And step 4 is where these amyloid beta oligomers activate kinases, particularly GSK3 beta and CDK5, which hyperphosphorylate the tau protein, causing it to detach from the microtubules. And what happens to these detached p-tau in the step 5? These detached p-tau aggregates into something called neurofibrillary tangles by collapsing axonal transport and also destroys synapses, and then basically ultimately killing neurons. And then it causes the cognitive decline, which we now recognize as Alzheimer's disease. All right. And now we'll have a look at the next slide. So here a critical nuance is being presented. Here the amyloid plaques alone do not kill neurons efficiently. The real executioner is the tau protein. So the relationship between the amyloid beta and tau is pretty much synergistic, and it's not sequential or linear. So a landmark experiment where they injected amyloid beta 42 fibrils into the entorhinal cortex of P301L tau transgenic mice, and the tau mutations alone produce modest NFTs, but with amyloid beta injection, the NFT burden increased over fivefold. And this tells us that amyloid beta amplifies tau pathology dramatically. And the key phosphorylation sites are SCR, which is serotonin, uh, 202, 212, threonine 214, and serine 422, which are driven by like at least five kinases, including GSK3 beta, CDK5, and DYRK1A. So once tau tangles form, they spread like a prion basically, by moving transneurally from the entorhinal cortex through the hippocampus and into the neurocortex in the pattern described by Bragg staging. So critically, it is the tau pathology, not plaque load, that best correlates with the cognitive decline in patients. And this is why dual targeting of both amyloid beta and also tau is the future of the Alzheimer's disease therapy. So now let's have a look at the neuroinflammation, which is a third pillar. So for many years, neuroinflammation was considered a secondary response in Alzheimer's disease, a bystander. But we now know it is a primary driver that amplifies the cascade. So there are three main glial mechanisms, and the first is the microglial activation. It is where these amyloid beta oligomers bind TLR4 and RAGE receptors on microglia and polarizes them into a pro-inflammatory M1 state that releases TNF alpha, IL-1 beta, and IL-6. And second is the NLRP3 inflammasome. It is where the amyloid beta fibrils trigger assembly of this intracellular protein complex, which leads to caspase-1 activation, which then leads to the maturation of IL-18 and IL-1 beta, which then causes the pyroptosis or pyroptotic death of neurons. And third is the astrocyte dysfunction. It is where these reactive A1 astrocytes lose their neurotrophic support functions and fail to clear glutamate, which then causes excitotoxicity. The genetic angle is pretty much crucial. It is where the TREM2, loss of function variants, triple the Alzheimer's disease risk by impairing the ability of microglia in order to phagocytose amyloid beta and also ApoE4, which then impairs this clearance basically. So now let's talk about the ATN biomarker framework and disease staging. So this ATN framework is proposed by Jack and his team, and it's updated by NIAAA in 2024. It is a modern framework for classifying Alzheimer's biology. Here A stands for amyloid beta. Here A captures amyloid pathology basically. Here is where the CSF, cerebrospinal fluid, amyloid beta 42 drops first, and PET amyloid scans become pretty much positive, and plasma p-tau 217 is emerging as a blood-based surrogate. And T, which is the tau, captures the tau angle burden. It is where the CSF tau 181 and p-tau 217 rise, and the flotta super PET tracer images tangles directly. And N stands for neurodegeneration. It is where the NFL in cerebrospinal fluid rises, and the MRI shows hippocampal atrophy, and FDG PET shows glucose hypometabolism in both temporal and parietal regions. And the 2024 NIAAA staging adds a pre-symptomatic stage 1, where the patients are biomarker positive but cognitively normal. And this is now the target window for prevention trials. And the key message out here is that the amyloid beta accumulation begins 15 to 20 years before the first symptom, which means our window for intervention is much earlier than we ever treated basically. And in order to study this pathway, we rely on 4 classes of experimental models. The first is transgenic mouse models, which are the workhorse. 3xTG carries 3 mutations, which includes APP, PSEN1, and MAPT, MAPT, producing both plagues and tangles. Here, 5xFAD uses FAD mutations and develops severe amyloid pathology within just 2 months. And these are powerful for drug screening, but have a critical limitation. And that limitation is that mouse ApoE differs fundamentally from the human ones. human ApoE4. And mice do not spontaneously develop NFTs without transgenic DAR mutations. And then we have cell culture models, which offer rapid throughput. And then we have iPSC derived neurons, which are the most exciting advance. Basically, patient derived fat neurons show elevated amyloid beta 42 is to 40 ratios, and also tau phosphorylation, which is basically recapitulating the human specific biology. And we have humanized chimeric models where the human iPSC derived microglia are basically transplanted into the mouse brains, which allows us for the validation of TREM2, and also ApoE4 biology in a human relevant glial context. And now let's talk about the brain organoids and organ and chip systems. So here, the single biggest failure in Alzheimer's drug development is the mouse to human translation gap. So more than like 99% of Alzheimer's disease clinical trials failed before 2021. And in large part because mouse models do not actually replicate human microglial biology, which is the ApoE4 dysfunction or cortical circuit architecture. So brain organoids are basically the self-organizing 3D structures that are derived from patient immunopotent stem cells and are beginning to close this gap. And CRISPR edited organoids with the APPV717 mutation spontaneously develop the amyloid beta aggregate and also tau pathology. And then we have fusion organoids, which combines both cortical and cholinergic regions, which allow circuit level analysis of memory pathways. And then we have organ on a chip systems, which go even deep down further, where the microfluidic chambers that separate neurons, astrocytes, and microglia in interconnected compartments that basically allow real-time imaging of amyloid beta transport and also glymphatic like clearance dynamics. So basically, all these platforms are already being used for first pass drug screening before doing any annual study out there. Let's move on to the next slide. So here we'll talk about multi-pronged therapeutic strategies. The complexity of the amyloid means that no single target will be sufficient. So that's why the field has converged on four parallel therapeutic fronts. First is the anti-amyloid immunotherapy. It is where the monoclonal antibodies that clear amyloid beta from the brain, which we'll cover in depth on the next slide. And second, we have anti-tau strategies. It is where the kinase inhibitors and tau targeting antibodies are there. And third, we have secretase modulators, particularly BACE1 inhibitors, that block the first step of amyloidogenic processing. And fourth, we have the most cutting edge gene and RNA therapies, which includes CRISPR and AAV gene mediated delivery. And the emerging consensus is that the biggest gains will come from the combination regimens targeting two or more pathways simultaneously, which Gorky has mentioned already, similar to how modern oncology moves from single agents to combination chemotherapy. So let's talk about the FDA approved anti-amyloid immunotherapies. So basically, this slide represents a genuine milestone in the history of neurology. For the very first time, we have disease-modifying therapies, basically the drugs that slows down the underlying biology of Alzheimer's and not just managing the symptoms. We have aducanumab in 2021, which was quite controversial because of mixed phase 3 data, accelerated approval, and also ultimately withdrawn its commercial use in 2024. It was historically important but clinically marginal, basically. And then we had lecanemab, or we can call Lecumbi. It changed the picture drastically. So that it targets soluble amyloid beta protofibrils, which is the most toxic oligomeric species, and it demonstrated a 27% slowing of cognitive decline over 18 months in 1795 patients. And FDA granted traditional approval in July 2023. And then we had donanemab, or otherwise called Kisunla, targets a pyroglutamate modified form of amyloid beta in mature plaques. It is Trailblazer ALS2 trial that showed 22% slower decline, and uniquely sufficient plaque clearance has been shown, which allows stopping treatment at a very early stage and a significant quality of life advantage, basically. And FDA approved this on July 2, 2024, which is very recent. And both of these drugs work by engaging FC receptors on microglia to phagocytose the antibody-bound plaques. And ARIA amyloid-related imaging abnormalities like brain microbleeds or edema occurs in like 21% of patients using this drug, and it requires MRI monitoring. So the success of anti-amyloid drugs has validated the cascade hypothesis and opened the door to the next generation of targets. In the anti-tau space, semorinumab and tilavolumab are the anti-tau monoclonal antibodies in phase 2 trials currently, which includes GSK3 beta inhibitors like titglusib, which aim to reduce tau phosphorylation at the source. For neuroinflammation, the NLRP3 inhibitor MCC950, it has shown strong preclinical results, and AL002C, a TREM2 agonist, is in phase 2, essentially trying to supercharge the microglial amyloid beta clearance by activating the same pathway that TREM2 loss of function variants disrupt, which I discussed earlier. And in the gene therapy space, ApoE42, ApoE3 CRISPR correction is currently being explored as a strategy to reduce the strongest sporadic Alzheimer's disease risk factor. And AI-driven drug discovery is accelerating everything. Basically, AlphaFold 2 guided secretase inhibitor design. We have GNN-based blood-brain barrier permeability screening, and ML prediction of amyloid beta aggregation inhibitors are all the active areas currently. So, despite the excitement, we need to be pretty much very clear-eyed about the challenges, and the most immediate clinical concern with anti-amyloid antibodies is ARIA. Roughly 1 in 5 patients developed MRI visible microbleeds or edema, which requires monitoring and sometimes treatment suspension. The magnitude of clinical benefit is modest. Basically, 27% of slowing of decline sounds impressive, but in absolute terms, if you think carefully on cognitive scales, it represents weeks to months of difference at max. And this raises real world benefit risk questions. Notably, the EMA refused lecanemab marketing authorization 2024 because citing insufficient benefit over risk for the broad population. The deeper problem is irreversibility. Once significant neuronal loss has occurred, clearing amyloid is like clearing flood debris after the flood has already damaged the foundations. So this drives a shift towards the prevention trials in biomarker positive asymptomatic individuals. So the future priorities are pretty much clear. We need to have earlier intervention, combination therapy targeting amyloid beta plus tau plus inflammation simultaneously, subcutaneous formulations for accessibility, and also nanoparticle blood-brain barrier delivery for next generation agents. So to summarize, the amyloid cascade pathway begins with a single misprocessed protein, which is the APP, which I have said at the start, which is the amyloid precursor protein, which then unfolds over decades through oligomerization, plaque deposition, tau hyperphosphorylation, neuroinflammation, and ultimately irreversible neuronal death. We have the most powerful experimental models in history, from 5x fad mice to patient-derived mouse organoids. And for the very first time, we have 2 FDA-approved therapies that actually show this, that actually slows down this process. But the work is far from over. The pathway is pretty much clear, and we have the targets identified. And what remains is the precision getting the right drug to the right patient at the right time, which means earlier diagnosis, better biomarkers, and combination therapies. So that's it for today's lecture. I hope you like this lecture, and if you have any questions, you can contact me at soy.bme@gmail.com. Thank you so much.
Thank you, Sohit. That was an insightful and interesting lecture. Now we welcome back Sanampreet Kaur for her next lecture, which is our last lecture. Her lecture is on landscapes of Alzheimer's disease. Sorry, her lecture is on transcriptomic landscapes of Alzheimer's disease. Welcome, Sanampreet.
Hello, everyone. Today, I'll be discussing the Alzheimer's disease from a transcriptomic perspective, focusing on how the patterns of gene expression shape the progression of the disease. For decades, the Alzheimer's disease has been defined by two pathologies: amyloid beta plaques and tau tangles. Amyloid beta is produced through the cleavage of amyloid precursor protein or APP, which accumulates extracellularly as plaques, while tau is a microtubule associated protein which becomes hyperphosphorylated and it detaches from the microtubules and aggregates into the intracellular tangles. These features are consistent, they're measurable, and they're also central to diagnosis and research. But when we look more closely, a problem begins to emerge. There is a disconnect between pathology and the clinical presentation. There are some individuals who carry a high burden of plaques, and yet they maintain relatively preserved cognition. On the other hand, there are others who are experiencing significant cognitive decline, but they have comparatively less visible pathology. So, plaques and tangles, while important, they do not fully explain when symptoms begin to emerge and how quickly the disease progresses or why there is so much individual variation. So, studying Alzheimer's disease with a focus on its end products, amyloid plaques, tau tangles, and accumulated proteins, has the obvious key limitations. It tells us what is present in the diseased brain, but it does not necessarily tell us what processes are driving those changes. So instead of asking which proteins are accumulated, we need to begin to ask a more fundamental question: that what changes in the cellular regulation leads to this state? And to study it, we need a tool that can capture the gene activity across the entire system. And that tool is transcriptomics. Transcriptomics is the study of the transcriptome, or the complete set of the RNA molecules that are expressed in a cell or a tissue at a given time. At a basic level, it contains two layers of information. It contains the DNA as a blueprint and the RNA as the activity. In Alzheimer's, the DNA sequence remains largely unchanged, but the patterns of gene expression are dramatically changed. By analyzing the RNA, transcriptomics gives us a snapshot of the cellular state. It allows us to see which genes are upregulated, which ones are downregulated, and which biological pathways are active or which ones are suppressed. So instead of focusing on one protein or one pathway, we can observe the system-wide changes across the neurons, the glial cells, and multiple brain regions at the same time. We can begin to track how gene expression changes from early to late stages, and how different cell types respond differently, and also how the disease unfolds as a dynamic biological process. And transcriptomic offers a functional readout of what the brain is actively doing at the molecular level. Now, the question is, how do we study the transcriptome? We actually use a powerful technique called RNA sequencing or RNA-seq. This method allows us to translate the molecular activity inside the cells into a quantitative profile of gene expression across the entire genome. And this process begins with the tissue collection, often from a brain-specific region such as hippocampus or cortex. And from this tissue, we isolate the total RNA, including the messenger RNA, which reflects the actively expressed genes. we first convert it into complementary DNA or cDNA using reverse transcription. Now, this complementary DNA is fragmented, amplified, and then assembled into a sequencing library. Next comes the high throughput sequencing, where millions of short sequencing reads are generated, and these reads are then aligned to a reference genome and then quantified to determine how frequently each gene appears. The result is a gene expression profile where each gene is assigned a measurable level of activity, and this allows us to compare conditions such as healthy versus diseased tissue, early versus late stage of Alzheimer's, and difference across brain regions and even difference across cell populations. What makes RNA sequencing powerful is that it tells us how active each gene is. There are two types of RNA sequencing, bulk and single cell RNA sequencing. In bulk RNA sequencing, we extract RNA from a piece of tissue and then we measure the gene expression as an average across all the cells that are present in that tissue. But the brain is a highly diverse cellular environment. It contains multiple cell types, each with a distinct function and expression profile. Like there are neurons involved in signaling, astrocytes which are supporting the metabolic and synaptic function, and then there are microglia which are important for immune response, and then finally there are oligodendrocytes which are responsible for myelination. So when we talk about bulk RNA sequencing, all of these signals are blended together, and this creates a major limitation. Cell-specific changes can be masked or they can be diluted, and in some cases, the opposing changes may actually cancel each other out as well. So we may risk missing the very signal that matters the most to us. To address this, we turn to single-cell RNA sequencing. Now, this approach isolates and sequencing RNA from individual cell, which allows us to identify cell type specific gene expression patterns, and it allows us to detect rare or transient cell states and also understand how different cell populations respond independently in disease. In Alzheimer's disease, this shift has been especially revealing. For example, neurons may show downregulation of synaptic genes, while microglia simultaneously upregulate inflammatory pathways. So these are fundamentally different processes that would be blurred or even lost entirely in bulk analysis. Although single-cell RNA sequencing tells us which cells are changing, but it doesn't tell us where those cells are exactly located within the brain. And in Alzheimer's disease, location matters a lot because pathology follows a distinct anatomical progression. And this is where spatial transcriptomics come in. It allows us to measure gene expression while preserving the spatial organization of tissue, effectively mapping the molecular activity directly onto the brain structure. In this technique, thin tissue sections are placed onto arrays which contain spatially barcoded probes, and RNA from each position is captured, sequenced, and then traced back to its original location. So instead of just asking what genes are expressed, we can now ask where they are expressed. This adds additional layers of information in Alzheimer's disease. First, there is regional specificity. We can track how molecular changes emerge and spread from early vulnerability in hippocampus to broader involvement in the cortical regions in the later stages. Second, there is localized dysfunction. Spatial transcriptomics reveals hotspots where microglial activation is elevated and inflammatory genes are upregulated, or where the synaptic genes are suppressed or downregulated. Rather than uniform damage, we see clusters of molecular disturbance. Third, there is the information about the disease progression or the disease propagation. By aligning gene expression patterns with anatomical pathways, we can finally begin to understand how changes in one region may influence the connected regions over time. And this brings us to global transcriptomic changes. When we compare gene expression profiles between healthy and Alzheimer's brain tissue, we don't see a random disruption. Instead, we see a more coordinated reorganization of the gene expression. Across studies, two major patterns consistently emerge. First, there is a broad upregulation of immune and inflammatory pathways. This includes increased expression of genes involved in microglial activation, cytokine signaling, and innate immune responses. Together, these changes indicate a shift towards a more chronic inflammatory state. At the same time, we observe a second and an equally important pattern, a downregulation of neuronal and synaptic genes. Genes which are associated with neurotransmitter release, synaptic vesicle cycling, and receptor function and plasticity are consistently reduced. Now, what's striking is that these two trends are not independent, they are occurring in parallel. So, as immune activity increases, neuronal and synaptic function declines. And this reflects a deeper biological shift. The brain appears to reallocate its resources from the connectivity and information processing towards defense and stress response. And this shift extends beyond the neurons and immune cells. So, transcriptomic studies also reveal disruption in the metabolic pathways, mitochondrial function, and broader systems involved in cellular maintenance and homeostasis. One of the most consistent findings across the transcriptomic studies of Alzheimer's is the upregulation of neuroinflammatory pathways. This is not a brief or a localized response. It reflects a sustained activation of the brain's innate immune system. And at the center of this process are microglia, your brain's resident immune cells. Under normal conditions, microglia survey the environment, they clear the cellular debris, and they also help to regulate the synaptic remodeling. But in Alzheimer's, the microglia undergo a profound transcriptional change. Transcriptomic studies have showed that there is an increased expression in the genes involved in cytokine signaling, including pro-inflammatory mediators such as interleukins and tumor necrosis factor. Also, there is this activation of complement system. While this mechanism is essential during development, in Alzheimer's it becomes excessive and even dysregulated, leading to inappropriate synaptic pruning. There is another important pathway involved here, that is TREM2, which is actually a receptor expressed on microglia. Upregulation of TREM2 is associated with a shift towards the disease-associated microglial state. Initially, this may be protective, which enhances the phagocytosis and the clearance of toxic material. But with chronic activation, microglia become persistently inflammatory and less effective at resolving the damage. And increasingly, they are disrupted to the neuronal function. So, what begins as a protective response gradually becomes maladaptive. Instead of restoring balance, chronic inflammation contributes to synaptic dysfunction, neuronal stress, and the progression of pathology. Alongside this rise in inflammatory signaling, transcriptomic studies also reveal an equally consistent pattern. There is downregulation of synaptic genes. These genes form the core machinery of the neuronal communication. They are involved in neurotransmitter release, like the synaptic vesicle proteins, like the components of SNARE protein complex. They are involved in receptor function, particularly the glutaminergic receptors like NMDA and AMPA receptors. And they also include the structural elements that maintain the dendritic spines and also the synaptic architecture. When the expression of these genes decline, the impact is profound. At the cellular level, the synaptic transmission becomes less efficient, and the brain's capacity for plasticity and adaptation reduces. And this is not confined to a single region. Transcriptomic data shows that the synaptic gene downregulation is widespread across multiple brain regions, reflecting a system-wide loss of interconnectivity. This provides a direct molecular link to the cognitive symptoms of Alzheimer's. There is memory impairment, which is driven by the disrupted hippocampal synaptic function. There is reduced learning capacity as the neuroplasticity mechanisms become weaker. And there is attention and executive dysfunction, which is linked to the cortical network disruption. Beyond inflammation and synaptic loss, the transcriptomic studies reveal another consistent feature of Alzheimer's disease, which is the downregulation of metabolic and mitochondrial genes. Neurons are among the most energy demanding cells in the body. They have to maintain the ion gradients for the action potentials, they have to take part in synaptic transmissions, and there has to be continuous protein turnover, all of which requires a constant supply of ATP, which is generated mainly through the mitochondrial oxidative phosphorylation. But in Alzheimer's, this energy system begins to falter. Transcriptomic data shows that there is reduced expression of genes which are involved in electron transport chain components, oxidative phosphorylation pathways, and ATP synthesis and mitochondrial maintenance. The result is a growing energy deficit. This reduced ATP limits the synaptic vesicle cycling, ion homeostasis becomes unstable, and neurons become much more vulnerable to stress and excitotoxicity. These metabolic dysfunctions do not occur in isolation. They interact with other changes that we have discussed. Inflammation increases the energy demand, synaptic dysfunction reduces efficiency, and mitochondrial decline limits the energy supply. And this creates a compounding effect. The brain is simultaneously using more energy while being able to produce less of it. The result is that system is under constant energetic stress. Another key dimension which is revealed by the transcriptomic studies is the disruption of protein homeostasis or proteostasis, the system that ensures the proteins are properly folded, maintained, and cleared. Under normal conditions, the cells maintain protein quality through tightly coordinated mechanisms. There are molecular chaperones which assist in proper protein folding. There is the ubiquitin proteasome system, which targets the damaged protein for degradation. And then there is the autophagy lysosomal pathways, which remove the larger aggregates and even the cellular debris. In Alzheimer's disease, these systems are not simply switched off, but they're dysregulated and they're constantly under strain. So transcriptomic data shows a mixed pattern. For example, the genes that are involved in unfolded protein response are often upregulated, indicating that cells are sensing stress, but the genes which are responsible for the efficient degradation and the clearance may be downregulated, or they may be insufficiently activated. So this creates a state of compensatory imbalance. The cells recognizes that the misfolded proteins are accumulating, and the cells attempt to respond, but the response is incomplete and overwhelmed. So because of that, misfolded proteins are not effectively cleared, aggregates begin to accumulate, and the cellular stress continues to escalate. From this perspective, the amyloid plaques and the tau tangles are not just isolated pathological features, they are actually the visible outcome of a broader failure in the protein quality control mechanisms. So far, we have looked at the global transcriptomic changes, but these changes are not evenly distributed across the brain. They're highly cell-specific. Single-cell transcriptomic approaches reveal that Alzheimer's is not just a uniform process, but it's a disorder that involves multiple interacting cell population, each with its own molecular response. So let's start with the neurons. Neurons consistently show downregulation of synaptic genes. Also, they show the reduced expression of genes involved in plasticity and communication. This reflects a loss of their core function, which is the ability to transmit and process information. In contrast, the microglia show a very different profile. They exhibit upregulation of immune and inflammatory genes. There is activation of pathways which are linked to phagocytosis and the stress response. This marks a shift from surveillance state to a more reactive and disease-associated state. Astrocytes, which normally support the neuronal function, also undergo a transformation. They increase the expression of reactive and inflammatory genes while reducing the roles in metabolic support and synaptic maintenance. Even oligodendrocytes, which are responsible for myelination, show an altered gene expression, suggesting disruptions in white matter integrity and support functions. Now, what is critical is that these changes are not independent. These changes form an interconnected network of interactions. Activated microglia influence the astrocyte behavior, reactive astrocytes impact neuronal survival, and neuronal dysfunction feedbacks into the system. Now let's talk about these changes in the individual cellular states in detail. In Alzheimer's, microglia do not simply become activated, they undergo a profound transcriptional reprogramming and they shift into a distinct cellular state known as disease-associated microglia or DAM. Under normal conditions, microglia exist in a homeostatic state. They are continuously surveying the brain environment. They clear the cellular debris and they support the synaptic remodeling. Their gene expression reflects a balanced and a more maintenance-focused role. But in Alzheimer's, this program shifts. Transcriptomic studies reveal that microglia move away from this homeostatic state and they transition into the DAM phenotype, which is characterized by upregulation of genes involved in phagocytosis, increased lipid metabolism and stress response pathways, and activation of immune signaling, particularly the TREM2 pathway. TREM2 plays a central role in driving this transition. Its activation enhances the microglial survival, it promotes the phagocytic activity, and it also facilitates the shift into this disease-associated transcriptional state. In the early stages, this response may be protective. Microglia attempt to clear the amyloid deposits, they try to remove the damaged cells, and they try to restore the tissue homeostasis. But with sustained activation, this state becomes dysregulated and microglia begin to maintain more chronic inflammatory signaling. They contribute to excessive synaptic pruning and they start releasing the factors which increase the neuronal stress. So instead of resolving the damage, they become part of a self-sustaining cycle of dysfunction. Also, the relationship between astrocytes and neurons undergoes a fundamental shift. And transcriptomic studies reveal this with striking clarity. So, astrocytes under normal conditions act as essential support cells. They regulate neurotransmitter levels, they maintain the ionic balance, they provide metabolic support to neurons, and they also preserve the synaptic stability. But in Alzheimer's, astrocytes transition into a reactive state, often referred to as A1 reactive astrocytes, and this state is characterized by upregulation of inflammatory genes, reduced expression of neurotrophic and supportive factors, and altered metabolic and homeostatic functions. This transformation does not happen alone. Signals from activated microglia, particularly the inflammatory cytokines, also drive the astrocytes into this neurotoxic phenotype. So astrocytes shift from being supportive partners and they contribute to a more hostile neuronal environment. Neurons also experience a fundamental shift. Transcriptomic data consistently shows that there is a downregulation of genes involved in synaptic transmission, reduced expression of plasticity-related genes, and decline in pathways that support communication and connectivity, as we have already discussed. This means that neurons are not simply dying, they're losing their functional capacity even before degeneration becomes apparent. And this brings us to the question of which models to choose to study these transcriptomic changes associated with Alzheimer's. To understand transcriptomic changes, we rely on the experimental models that allow us to study mechanisms under controlled conditions. But there is a catch. No single model fully captures the complexity of the human brain. Instead, we use complementary approaches, each offering a different window into the disease. The first approach is transgenic mouse models. These mice are engineered to express human genes associated with Alzheimer's, such as the amyloid precursor protein and tau mutations. This allows researchers to track disease progression over time and examine how gene expression evolves with pathology. These models are especially useful for studying the causal mechanism and also for performing the longitudinal analysis and for testing the potential interventions. The second approach is the iPSC-derived neurons. Here, the somatic cells from patients are reprogrammed into induced pluripotent stem cells, and these then differentiate into neurons. And this provides a patient-specific genetic background and insights into the disease processes at the cellular level. These models are valuable for studying individual variability and for studying the early molecular changes and for studying the human-specific gene expression patterns. The third approach is the brain organoids. And these are three-dimensional structures which are derived from the stem cells that mimic the aspects of the brain organization. They allow us to study cell-to-cell interactions and the early developmental and disease-related processes and also more complex tissue-like environments. And each of these models captures a different aspect of the disease. Together, they act like complementary lenses which help us to approximate the transcriptomic landscape of Alzheimer's from multiple different angles. While experimental models have provided the valuable insights into Alzheimer's transcriptomics, they also come with important limitations which we need to discuss. No current model fully captures the complexity of the human brain or the full trajectory of the disease. There is this issue of cell type diversity. Like the human brain contains an extraordinary range of different cell types, like the neuronal subtypes, the glial populations, and each of these cells also have the region specific cellular identity. And the models, like the mouse models, they cannot reproduce this diversity. Second, there is this issue of temporal complexity. Alzheimer's develop over decades in humans. In contrast, in mouse model, the disease progresses over months. And the cell culture system, they can capture only the acute and the early stage conditions only. This makes it difficult to model the long-term adaptations and the progressive network dysfunction and the late-stage disease dynamics. So there is this incomplete temporal information which can be provided by the models. Third, there is this incomplete representation of molecular pathways. Many models are built around the specific features such as the amyloid accumulation or the tau pathology. But Alzheimer's involves coordinated changes across multiple different systems. It involves changes across immune responses, the metabolic pathways, the synaptic function, and transcriptomic changes are only partially reproduced. Fourth, there is this challenge of translation to human diseases. Many therapies that appear effective in the preclinical models fail to show the benefit in the human trials. And this highlights a critical gap. What works in a controlled experimental system does not always reflect the biological reality of the human disease. So while these models are essential, they are inherently simplified representation and they each capture only a part of a much larger system. And this further reinforces the central idea that Alzheimer's cannot be fully understood through any single model or any single approach. It requires integration across multiple systems, especially incorporating the human-derived transcriptomic data. Across this presentation, we have moved from the traditional views of Alzheimer's to a more integrated and transcriptomic perspective. Historically, Alzheimer's has been framed around single pathological features, the amyloid plaques and the tau tangles. While these are important, they represent only a part of the story. Transcriptomic studies reveal a much broader and a more dynamic picture. So the broader realization is this: Alzheimer's is not just a disease of accumulation, but a disease of misregulated biological programs which are unfolding across a complex cellular environment. And transcriptomic gives us a way to read those programs as they change over time. Thank you.
Thank you, Sanamjeet. As we now conclude today's session, I would now like to invite two distinguished researchers who will be sharing valuable academic and mentorship insights for students aspiring toward research or PhD opportunities. We have Dr. Brooks Robinson from UCCS, and we have Professor Alfredo Alguezi from the Department of Biology at the University of Puerto Rico. We are honored to have both of them share their insights with us. Before we move into these mentor talks, I would like to sincerely thank all our student, speakers, mentors, and audience for making today's session meaningful. We look forward to continuing this journey in the upcoming session, which is on 7th of May, where we will be exploring disease biology, drug discovery, and interdisciplinary approaches in life sciences. With that, we now conclude today's session, and I invite all of you to watch the mentor talks. Thank you, everyone. Thanks.
What if I told you that getting into your dream PhD program wouldn't come down to your grades or even your research experience, but to how well you play the game? Hi, my name is Brooks Robinson, and I've been on both sides of the table. I've applied to PhD programs myself, I've also reviewed PhD applications, mentored students, and watched brilliant minds get overlooked because they didn't know the rules of the game. Let's face it, most students don't fail because they're not smart. They fail because no one taught them how the system really works. I'm part of a team of professors, scientists, and experts led by Dr. Sukant Khurana. We've helped students from all over the world get into top PhD programs in Germany, France, the Netherlands, and beyond. We know what works: how to write a letter that lands an interview, how to email professors and get responses, and how to write a CV that helps you stand out from the hundreds of other applicants. This is not guesswork. This is strategy from the other side of the table. Instead of stumbling through the process, learning from your mistakes, learn from ours. We've reviewed hundreds of PhD applications and helped shape even more. Why go it alone when you can be guided by the people who make the decisions? If you're serious about a PhD in Europe, stop guessing. Email us at contact@ioncurerx.com with your CV and goals, and let's build you an application that gets results. Because success isn't just about hard work, it's about smart work with the right team. Join the students who've made it because they have the right mentors. You could be next.
Hello, I'm Alfredo Getzi, professor and director of a graduate program, and someone who has spent years reviewing PhD applications from around the world. Let me tell you something honestly. Far too many brilliant students don't make it through the door, not because they lack potential, but because of avoidable mistakes in their applications. Here are just a few common issues I see again and again. A generic motivation letter that could apply to any lab, anywhere. A CV that reads like a shopping list, not a story of academic growth. Students who fail to connect their past work to the lab they're applying to, and surprisingly often, poor communication with the potential supervisors. These things may seem small, but trust me, they are deal breakers. But here's the good news. You don't have to figure this out alone. I work closely with Dr. Sukant Khurana and an international team of professors and scientists. We have sat on admissions panels. We've guided hundreds of students, and now we're here to guide you. We understand what admissions committees are really looking for, and we are ready to help you present your story in the best possible light, tailored, strategic, and authentic. So, if you're serious about pursuing a PhD, especially in Europe or North America, don't leave it to trial and error. Reach out to us at contact@ioncurerx.com. Let's work together to make your application not just good, but unforgettable.